Watch this video before your next professional exam. Let us talk about the pathophysiology of sickle cell anemia. Of course, you know that sickle cell anemia is as a result of combination of SSG from the father and the mother. Let us dive into the root cause of this sickle cell anemia. How did this sickle cell anemia come about in the first place? Because it's actually a genetic disorder that is transferred from the parents to the offspring, and so it goes on and on. Now, there are what we call the bases of the DNA or the DNA bases, which include adenine, thymine, cytosine, and guanine. These bases are arranged in sequence that form instructions for making proteins like hemoglobin. These bases are also read in codons. Each codon specifies or produces one amino acid. Examples of this codon include GAG codes for glutamic acid, while GTG codes for valine. So if there is a change in any of the base structure, it actually changes the kind of amino acid that will be produced. What happens in normal adult hemoglobin? In a normal adult hemoglobin, the beta globin chain has a codon for GAG, which is glutamic acid. Now, glutamic acid has the following properties. It is hydrophilic, meaning that it's water-loving. Secondly, it makes the hemoglobin molecules to be separate, because in a sickle cell anemia, the hemoglobin molecules are clumping together, making the cell to become rigid. Now, lastly, it makes the red blood cell to become flexible and round. That is why your red blood cell can easily pass through a narrow opening or a narrow vessel. But in sickle cell anemia, these rigid cells, which are not flexible and not round enough, rather they have a crescent shape, cannot pass through these narrow vessels inside of the body, which causes vaso-occlusion, making the red blood cell to become clutched in a particular area of the body. And that is why the sickle cell patient will be feeling pain in that particular area due to vaso-occlusion. Now, in sickle cell, a single base mutation occurs, meaning that adenine will be replaced with thymine, which also affects the codon. The GAG, which is glutamic acid, will now be replaced with GTG, which is now valine. In all these mutations that has occurred, they call it a point mutation. Now, how is valine produced? Valine is produced because there is a change from adenine to thymine, which also affects the codon, changing it from GAG to GTG. GAG is for glutamic acid, which is that of a normal hemoglobin, while GTG is for valine, which is that of a sickled hemoglobin. This alteration in hemoglobin is called hemoglobin S. A normal hemoglobin is called hemoglobin A, while a sickled hemoglobin is called hemoglobin S. Now let's talk about the effect of valine. The effect of valine on hemoglobin is critical because valine is hydrophobic. It makes the hemoglobin to attract each other. Secondly, it makes it to polymerize under low oxygen. And lastly, it makes the cell to become rigid, or perhaps forming rigid fibers inside of the cell. These rigid fibers makes the red blood cell to be distorted or sickled. Secondly, it causes less flexibility, that is making the red blood cell to lose their flexibility, because a normal red blood cell is meant to be flexible so that it can easily pass through narrow vessels which may be found in the leg. Now, thirdly, it makes the vessel to become blocked, or perhaps we call it vessel blockage. And lastly, it causes cell destruction, which is called hemolytic anemia, because sickled blood cells don't reach the normal lifespan of a red blood cell, which is 120 days. A sickled red blood cell destroys within 10 to 20 days. That is why people who are sickled do lose blood due to hemolysis. And now, one of the cardinal signs of someone that has sickle cell is their body appears yellow due to the excess secretion of bilirubin. How did this bilirubin come about? In a sickled blood, it loses the ability to reach 120 days as a lifespan. Rather, it becomes destroyed at 10 to 20 days when it's being produced. Now, one of the products of destruction of red blood cell is bilirubin. So when these sickled blood cells are being destroyed often, this bilirubin will be produced in excess, and the liver cannot walk up to the amount of the bilirubin that will be produced. It begins to secrete this bilirubin inside of the bloodstream, which makes the body, the extremities, the eyes, the mucus to appear yellow. That is why you can easily note if a patient has a sickle cell. This entire cascade started from a one base letter change in DNA. That is, GAG was changed to GTG. That is, the adenine was replaced with thiamine. Now, when does this sickled hemoglobin manifest? It doesn't manifest immediately after birth. When a baby is given birth to, the baby still has hemoglobin F, which is called the fetal hemoglobin. After like 5-6 months after birth, some factor can trigger the modification of HbF to HbS, that is from fetal hemoglobin to sickled hemoglobin. The factors include during infection, during low oxygen, dehydration, respiratory acidosis. These factors can trigger the modification of HbF to HbS, and that is when the baby will start, or the child or infant will start manifesting with the signs of a person that has sickle cell anemia. Now, this sickle cell anemia can be prevented. Before you get married to a person, you have to confirm or check your genotype. That is, if you are AA, AS, or SS. AS is not meant to marry SS. SS is not also meant to marry AS, because this can lead to the birth of a baby with SS. So in all this that I've said, you might feel like this is not necessary to go for a test, but it is, because it will affect the unborn child. The baby is going through all this casket because of something that could have been prevented by a man and a woman. If you have a question as regards to the sickle cell anemia, you can drop it in the comment section. I promise to reply to them. Thank you.